There's another microphone.
Hello? Test.
Perrier, can you hear me? Perrier, can you hear me?
Yeah. Perrier.
What do you got in your mouth? Sweet.
Okay.
So that'll be okay.
Yeah. This.
Let's turn it off now.
Oh, whatever you can. Yeah.
The crowd is pretty spread out. stragglers way over there to the east. We're going to have to pick on you guys because you're kind of aberrant. Are you worried about it? Be worried. Well, I'm truly happy to introduce my colleague and friend, Hélène Cote, and also this guy, Zhe Li, who will be presenting at today's summer research day for CBR. Hélène did her graduate work here at University of British Columbia with our first Center for Blood Research director, Ross McGillivray. So she's a coagulation biochemist. That's why I like to have her on my committee, so she can bug my students really well. She learned all about molecular biology and protein chemistry in Ross's lab, and then she went south to train as a postdoctoral fellow in Earl Davies' laboratory, where she learned more about coagulation biochemistry and also much more about molecular biology and molecular biochemistry. She then came north again, back to Vancouver, where she did postdoctoral fellow training further at the Center of Excellence in HIV and AIDS at St. Paul's. And so I think that this is a really interesting career path where she's bringing together molecular biology training into a new field in her life, and that is pathology, or pathogen. And so she's going to tell us more about that in HIV and possibly aging. I'm not sure what you're going to, and aging. I got it right. She's going to tell us about HIV and aging and how molecular biology fits into all of that picture. So I'm going to be quiet now and let Hélène take over. Thanks very much, Hélène, for joining us.
Okay. Well, thank you, Ed. Yeah, I took quite a turn, and I'm definitely not doing blood coagulation anymore, and some molecular biology, one would say. But today, we chose to do an unusual presentation, mostly rather than just presenting our hypothesis-driven research and our results, we wanted to talk about unexpected things we encountered and rabbit holes we went down trying to follow these. And so since these are real in research, we thought, why not share and you take what you may from this. So the title of the talk is Unexpected Turns Encountered Studying HIV and Aging, and also, preterm birth. And before I go further, I'd like to acknowledge that we are, all our research has taken place on the unceded ancestral traditional territories of the Cote Salish people. And this is also where we are meeting today. And we honor the Squamish, the Musqueam, and the Tsleil-Waututh, acknowledging that we are uninvited settlers on their beautiful land. I also wanted to have a note on language that in this presentation, we use the term women, and we usually are referring to people assigned female sex at birth with a uterus. But we acknowledge that not all people with a uterus identify as women, and not all people who identify as women have a uterus. And you will see that sometimes, as I said, the word female is used, sometimes the word women, sometimes both appear on the same slide. And this is really in part due to existing literature that has been conflating the two, even though they don't mean the same. So as an outline, first I will try to give you a brief overview of why we're interested in studying aging in people living with HIV. And I will describe cohorts we have been using over the past almost 20 years, and then present 3 vignettes, each showing a bit of an unexpected observation and what we did about it. The first will have to do with placenta mitochondrial DNA mutation in the context of HIV pregnancy and preterm birth, actually the first 2, and they will be presented by Zhe. And the third will have to do with the risk of cardiovascular events in women living with and without HIV, and well, how much is HIV playing a role in that? So why are we studying HIV and aging? Mostly because people living with HIV experience accelerated aging, which manifests as a shorter lifespan, shorter health span, so more comorbidities and aging-related diseases earlier in life. Notably, women living with HIV also have a 2 to 3 times higher risk of experiencing a preterm delivery, which is a risk equivalent to a 48 to 50-year-old pregnant woman. And it's not clear why that is, and that's what we're attempting to elucidate. I want to introduce briefly the hallmarks of aging. When this wheel was first published, there were, I think, 9, and now it's been expanded. And these are all, hallmarks or markers or functions that are affected as we age and really drive the aging process. And we will not touch on all of them, but some of them today. And when we are studying aging, we are considering most of these as best we can. So why is it that people with HIV experience accelerated aging? It's probably multifactorial. Clearly, is it HIV? HIV causes chronic immune activation. So even when people are on antiretroviral therapy, have undetectable viral load, they continue to have chronic immune activation and chronic inflammation at a low level, which is believed to probably contribute to an acceleration of aging process, as inflammation was one of the hallmarks of aging. It could be their HIV meds, because these they take a cocktail of at least 2 to 3 medication, and these medications have off-target effects. Some of them can affect the mitochondria. Some of them may affect telomerase, which indirectly may affect telomeres or stem cells, etc. So that could be it. And then it could be everything else in their life. It could be that they are more likely to have other chronic and latent viruses. They're more likely to maybe have substance use issues, because that may have been how they got their virus. It could be an issue with sex and gender, but it could also be to do with social determinants of health. And by that, we mean, your access to healthcare, your access to education and education attainment, your environment, your social support, your experiences of racism, of stigma, of your income, are you living in poverty or not, your housing security, your food security. All those things are stressors on the body in the end, and it's very plausible that they would affect aging. And typically, people living with HIV may have more adverse social determinants of health than the general population. So going back to that wheel of the hallmarks of aging, I'm pointing out here some of the ones that we know about. So we know that the virus itself causes chronic inflammation. It affects telomere. People with HIV will have accelerated immune cell division, and the telomere are shorter. It affects the mitochondria. People with HIV have lower mitochondrial DNA content, maybe part of the wasting process they experience if they're not treated, and so on. And then the medication themselves will affect the microbiome, will affect telomere through perhaps telomerase inhibition, and are known to affect mitochondria, so could affect mitochondrial function. So when we look at the life cycle of HIV briefly, I wanted to introduce why it would make sense for us to look at mitochondrial DNA. Almost everybody treated for HIV receives what we call a nucleoside reverse transcriptase inhibitor. And those are nucleoside analogs that can basically fool the polymerase gamma in the mitochondria and get incorporated in replicating mitochondrial DNA. So because this enzyme has a poor repair mechanism and is very error-prone, we hypothesized that this may increase mutation in the mitochondrial DNA. So the drugs that are in this position of the life cycle and are meant to prevent the reverse transcription of HIV RNA into cDNA prior to integration in the genome, they would also affect the polymerase gamma, the mitochondrial polymerase, and cause mutation. But they will also affect the telomerase, because telomerase really is a reverse transcriptase that is meant to replicate the ends of our chromosome. So moving on to the various cohorts that we've been studying for almost 20 years, as I mentioned. So the first two I'm presenting, for each of these cohorts, I wanted to just briefly mention when these cohorts took place, where, who they enrolled and recruited, how many participants they included, and what kind of biospecimen they collected. The first two are Karma Preg and EPIC, and I apologize, I don't know exactly when EPIC took place. That cohort is by Wendy Robinson, and she was collecting placenta primarily from preterm birth to try, and she was doing an epigenetic studies on placenta. Karma Preg enrolled women throughout their pregnancy, multiple visit, extensive data collected, as well as extensive collection of biospecimen. This was followed by Karma, or around the same time we did Karma. Instead of pregnant women, Karma was children and adults either living with HIV or controlled, not living with HIV, or exposed to HIV, meaning children born to a mom living with HIV. We also have, and this was in 4 different cities in Canada, over 1,000 participants, and all these specimens are in my freezers. Then we have the CLSA, which is the Canadian Longitudinal Study of Aging. I'm not an investigator on that study. I'm actually a participant. Just had my 3rd or 4th visit last week. It's been ongoing since 2010. It's throughout Canada, 50,000 people, almost randomly selected between the age of 45 and 85, and we are not accessing their specimen. More recently, we merged the Karma cohort with another cohort called CHIWOS, and we call it the British Columbia Karma-CHIWOS collaboration. It enrolls cis and trans women over the age of 16. Our target is 700 participants, and we've almost reached it, and we collect a number of specimens. And finally, more recently, 2 other studies, REDOSE and BCC3 reaching further. REDOSE is a study that is looking at the side effects of HIV medication. We're teaming with them because BCC3 was enrolling women, REDOSE was enrolling men, so that allowed us to do sex-based analyses. And then BCC3 reaching further is going into the trans and gender diverse population, so trans men, trans women, and non-binary participants. And that one is just about to start enrollment. So 3 of these studies are what we call community-engaged research studies, meaning that we work directly with the population, either people living with HIV or people who are in the trans and gender community themselves, or both. And they inform our studies from the get-go, from the research questions, from the designing of our survey. They pilot it with us. They are hired and work to administer the survey to our participants. They contribute to interpretation of data as well as presentation and publications. Today I'm going to be talking about data coming from Karma Preg and EPIC in the Verweerge Zeshow, and then I will follow with data coming from the CLSA and the BCC3 afterward. So our first vignette, I'll invite Zeshow, who's a PhD candidate in my lab, who threw his back a few days ago and is having trouble just standing. He's investigating whether preterm birth is associated with increased somatic placenta mitochondrial DNA mutation, and whether or not this is exacerbated in women living with HIV, but he's going to tell you about a couple of unexpected turns.
Thank you, Ellen, for the introduction. So firstly, I would like to give a brief background of mitochondria. Mitochondria is a dynamic cellular organelle. It has many important functions in the cell. Mitochondrial DNA, unlike nuclear DNA, it's a circular genome. It has high copy numbers. Normally, it could be from 100 to 10,000 copies per cell. On the right side of the slide, it is a genome map of human mitochondrial DNA with the information of functional regions, and this map is provided by the MetaMap database. So because of its high copy number, when we talk about mutations or variants in the mitochondrial DNA, we define two types of mutations. If variants or mutations present equal to or less than 2% of its genome is called heteroplasmy. If mutations present less than 2% of its genome, it's called somatic mutations. These are mutations with low frequency, and we can also call them rare mutations. So on the bottom left, there are just two diagrams showing the concept of heteroplasmy and somatic mutations. So to identify the low-frequency somatic mutations, we use the ultra-rare mitochondrial DNA mutation detection assay. Briefly, we used two identifiers, MID multiplex identifier, which can indicate the sequencing rates from the same participant, and unique molecular identifier, UMI, which can recognize the rates from the same mitochondrial DNA molecule. So the targeted sequencing, the targeted sequence in the mitochondrial DNA control region was tagged by the MID and UMI. Then we amplify them and we send them to sequencing. Once we have the sequencing rates back, we can group our rates into the families by using the MID and UMI. So for each family, we did the error filtering step. So we excluded the families with less than 5 reads and the mutations present in less than 75% of the reads in that certain family. Those are assuming to be the sequencing or PCR errors. And finally, we can generate the consensus sequence and identify the low-frequency mitochondrial DNA mutation. So the unexpected observation during our study is that the placenta DNA coming from epi cohort has systematically higher somatic mutation burden than placenta DNA coming from gamma preg cohort. The main difference, the main potential difference identified was the placenta DNA extraction method. The gamma preg uses a QIAGEN column kit, while epi cohort uses the sorting out method. So it drove us to a question that, does DNA extraction method matter for the rare mitochondrial DNA mutation detection? Because as you know, in our lab, we used to don't pay attention to which DNA extraction method is used, because DNA is quite stable and it's easy to be transferred and started with in the research studies. In the literature, the low frequency or rare mitochondrial DNA mutations are increasingly studied in many research fields, including neurodegeneration, cancer, aging, and mitochondrial disease. So there are molecular barcoding-based sequencing methods which can identify the low-frequency mitochondrial DNA mutations due to sequencing or PCR errors. But if artifacts were introduced during the DNA extraction step, these artifacts cannot be distinguished from the true mutations. So this led us to investigate the relationship between the DNA extraction method and the low-frequency mitochondrial DNA mutations. So here are our experimental design and study workflow. We used two types of specimens, placenta and PBMCs. For each specimen, it was divided into 2 equal parts for parallel DNA extraction, QIAGEN and sorting out. The main difference between those two methods is that the QIAGEN is coming with a commercial kit. It uses the silicon column to isolate the DNA, while sorting out has its own lysis buffer, and it also includes a protein precipitate step using high salt. After the extraction, we used our ultra-rare mutation detection method to detect the low-frequency mitochondrial DNA mutations. So here are our results. They are results from our placenta specimens extracted by 2 methods. And in the figure, we can see there is increased G2A mutations in the placenta extracted by the sorting out. And based on that, we believe the difference of the two cohorts we saw previously is driven by the elevated G2A mutations. We also did the comparison of somatic mutation burden in the placenta. And in the panel A, we can see there is increased somatic mutation burden of the placenta samples extracted by sorting out compared with QIAGEN. For the panel B, when we excluded the G2A mutations from the mutation burden calculation, the significance disappeared. However, in panel C, if we just look at the G2A mutation burden, we can see the significant difference once again. So all these results indicate that the increased mutation burden in sorting out extracts is primarily driven by the G2A mutations. We were thinking that whether the increased G2A mutations are related to the background noise. So we did the spectrum analysis for the filtered out sequencing of PCR errors. And here in this figure, we saw there's no significant difference between the samples extracted by two methods, which means the elevated G2A signals cannot be explained by the sequencing of PCR background noise. So as we previously mentioned that the sorting out method includes protein precipitation step using high salt. And in our protocol, we incubate the tissues in the high salt for 10 minutes. So could the increased G2A mutations be related to the high salt exposure? To investigate that, we studied the G2A mutation burden over the time course of 500 to 300 minutes in the high salt. And here are the results. For the figure on the left, all the dots belonging to the same placenta are in the same color, and they are connected by lines. And here, in this figure, we can see there's no consistent pattern of the G2A mutation burden over the high salt exposure. For the figure on the right, we also use the Loi's regression to visualize the trend of G2A mutations over high salt exposure. And here in this figure, we didn't observe any increase or decrease trend over the high salt exposure. And we also did some statistical analysis, and the median between time points is not significantly different. So based on that, the longer high salt exposure has no effect on the G2A mutations. Next, we were thinking, is this something strange specific to the placenta tissues? Because placenta is a dying tissue. It's different from other tissues. So we have the similar comparison of the somatic mutation burden in the PBMC extracted by two methods. And here we found a similar pattern of the PBMC as we saw previously in the placenta specimens. And specifically, I want to point out in panel C, there is an increased G2A mutation burden in the PBMC extracted by the sorting out method compared with QIAGEN. So now we know it's not tissue specific. We want to look a little bit further on where does the G2A mutations come from. So we did the experiments to swap the lysis buffer between two extraction methods. And here are the results. So in all those four panels, there are four groups in the comparisons. Q is a group using the standard QIAGEN protocol. Q plus SOLB is a group using the QIAGEN protocol with sorting out lysis buffer. SO is a group using the standard sorting out protocol, and SO plus QLB is a group using the sorting out protocol with QIAGEN lysis buffer. For the panel A and B, we compared the total somatic mutation burden. And in panel A, we can see, although the p-value didn't reach to the significance level, the median of group Q plus SOLB is higher than the median of group Q. And when we look the data a little bit further, we also saw some samples with the red color dots. And for those samples in the group of Q plus SOLB, there are a bunch of G2A mutations which has a frequency above 2% cutoff, and those mutations are removed from the mutation burden calculation. For the panel B, we can observe there is an increased somatic mutation burden in the sample extracted by the standard sorting out compared with the sample using the sorting out protocol with QIAGEN lysis buffer. And for the panel C and D, we observed a similar pattern as we saw in panel A and B. And the panel C and D are the comparison of the G2A mutation burden only. So based on all these figures, the lysis buffer chemistry plays a critical role in the increased prevalence of G2A mutations. We also did the experiment called the sequential extraction experiment. For example, we extract our placenta specimens using the QIAGEN method to generate the DNA. Those DNA was re-extracted by the sorting out method and vice versa. And here in this figure, we compared the G2A mutations burden. And in the group between Q and the Q2SO, we can see there is no significant difference. And also in the group between SO and SO to Q, there is no significant difference as well. So based on these results, the sequential extraction experiments has no significant effect on the G2A mutations we saw previously. In conclusion, the increase in the G2A mutations is not a bias of sequencing or PCR errors. It's not related to the high salt exposure, and it's not tissue specific. However, it is associated with the tissue lysis condition using the sorting out buffer. For the take-home message of this study, the DNA extraction is an important pre-analytical determinant of rare mitochondrial DNA mutation detection, and it may have some implications to the nuclear DNA studies. Secondly, the DNA extraction methods should be reported in the study of rare mitochondrial DNA mutations. In the literature, there are about 60% of low-frequency mitochondrial DNA mutation studies failed to report this DNA extraction method in their method section. For the second linear, it is regarding to the study of investigating the possible association between placenta hydroplasmy, HIV, and preterm birth. Edward and I contributed into this study, and this study uses the placenta specimens from both Kamma-Pratt cohort and EPI cohorts. So to study the hydroplasmic mutations in mitochondrial genome, we used the whole mitochondrial genome sequencing method. Briefly, we amplified the mitochondrial genome with 2 overlapping fragments, and then we purified the applicant by the gel electrophoresis. After that, we do the gel extraction. We just extract the applicants back from the gel and send them to sequencing. Finally, we did some bioinformatics analysis to identify the hydroplasmic mutations, which are mutations of the frequency equal to or above 2%. Here is a simplified demographic table of 104 placenta specimens used in this study, and it includes 54 preterm births and 50 term births. In the table, I also list several variables, including maternal age, HIV exposure, ethnicity, and infant sex. I won't go to the details about the value number of these variables, but I want to say for all the variables, the p-value is not significant, and which means for all these variables, there is no significant difference between the group of preterm births and term births. So here it is a heat map of all hydroplasmic mutations we detected in the mitochondrial DNA genome. And each hydroplasmy has been colored into the reports of pathogenicity in the MetaMap database. And on the y-axis, you can see all the placenta specimens has been grouped by the birth status and HIV exposure. For the top x-axis, all the heteroplasmy are grouped by the control region or non-control regions in the mitochondrial DNA genome. So here, I think it's really obvious that we can see a green mutation which present at almost every single placenta specimens we have in this study. And we did a little further research on it, and it's, this mutation is 310T2C, and it has almost 40% frequency in the gene bank, and it's a haplogroup. So an important thing I want to mention based on this figure is that there are 3 mutations colored in red, which is the confirmed pathogenicity in the MitoMap database. And for these 3 confirmed pathogenic mutations, 2 of them are coming from EPIC, and one is coming from Kamaprag cohort. And for those 2 of EPIC, one has about 7% frequency, and it's located in the protein coding region. It's related to the exit syndrome. For the other one of EPIC, it has 5% frequency, and it's located in the ribosome RNA region. And this mutation is related to the hearing loss or the autism spectrum disorder and intellectual disability. For the one of the Kamaprag, it has 10% frequency, and it's located in the protein coding region. And this mutation is related to the Lee disease, Miller syndrome, or the labral hereditary optic neuropathy, Miller's overlap syndrome. Since the EPIC cohort was anonymous, we are not able to do the further follow-up step to those 2 pathogenic mutations. But for the one in the EPIC, we can do something about that. So for the observation of pathogenic mutations or variants which can be related to the mitochondrial disease, it would trigger a protocol for potentially clinically actionable incidental research funding. Firstly, we need to repeat the sequencing of the placenta tissue to confirm that pathogenic mutations. If it is confirmed, we would investigate other available infant tissues, for example, blood or cold tissues. And we would like to also investigate whether the maternal tissues carry that specific mutations, since the mitochondrial DNA mutations are maternal inherited. Once they are confirmed, we're gonna consult with the clinician who has the expertise of mitochondrial disease. And after that, they're gonna tell us whether the mutation frequency is needed to be followed up, and if it's clinical warranted, we're gonna track back the patient or the participant, and in this case, we're gonna contact to the pediatrician to do that. And also as a note, this placenta was collected in 2009, and the mitochondrial DNA analysis was done in 2024. So here is a multi-tissue analysis for the step 1 to 3. And here, for the mutation names on the y-axis of the heat map, I colored them by the reports of pathogenicity reported in the MitoMap database. And for each heteroplasmy, they are colored by the frequency level. And for the x-axis, we can see we sequenced the several infant tissues and also 2 maternal tissues. So here in the red box, this mutation colored in red is the mutation we saw before. And we can see this pathogenic mutation is detected in all infant tissues at a similar frequency level, around 10 to 11%. And it's not detected in the maternal tissue, which indicates that there is a possibility of the de novo mutations happen early in the embryonic development. So now we have finished our research detection and the internal review steps. Then we consult to the mitochondrial DNA expert, and they mentioned to us these pathogenic mutations raise the concern for the mitochondrial disease. Although it has a relatively low frequency, like 10%, the risk may be influenced by the mitochondrial toxins, for example, smoking. So it is needed to be followed up. And then we contact the pediatrician, and the reply from that is the patient is 17 years old now, the patient is male, so there's no chance of the mitochondrial DNA mutations inherited into next generation. And currently, the patient has no symptoms. So based on that, the decision would be there won't be the further genetic testing pursued at this time, and they would open the file for the future reassessment if health concerns arise. So as the benefit of this follow-up on the incidental funding, in this case, the youth has been reconnected with the healthcare, and the pediatricians are happy with that because they also found other health issues of the patient they need to pay attention to. And the patient has been received the practical mitochondrial health advice, for example, avoid smoking, and the patient has been linked with the specialist if the further follow-up needed. Yeah, then I will pass the slides to Alan again. So for the following content. Thank you.
Sorry. All right, thank you. So vignette number 3, we're taking a different turn. This is work that was done by Tatiana Povshedna. We call her Tanya. She could not be here today because she has started her residency and it's keeping her extremely busy. This, as I mentioned, will use data from the BCC3 and CLSA cohorts investigating estimated risk of cardiovascular disease in women living with HIV. And why are we doing this? Well, as I mentioned earlier, people living with HIV have a shorter life expectancy, but our study is studying females, as you know, or women, actually, cis and trans. And if you look worldwide, it's well established that women live longer than men. And in this graph, it shows you the life expectancy of women on the y-axis and the life expectancy of men on the x-axis. And the line is difficult to see, but I have amplified it here. This is the line if both male and female had equal life expectancy. So you can easily see that the women are mostly all above the line. I was disappointed that Canada was not on the map, so for your benefit, I added it. We are up there. So in Canada, women, I think, have a life expectancy of 84 years, and males, 79.4, something like that. So almost 5 years difference. And when we look at people living with HIV, this is where their life expectancy is. So 2 things. In BC anyway, this is data specific to BC. So you can right away see that both men and women have a much lower life expectancy, but women more so than men, because this is the only point that is below the line. So women have a life expectancy, I think, almost 7 years shorter than males living, or men living with HIV. So 61 and 68. And so this is what triggered our study of women living with HIV, trying to understand where is this coming from. It's also established from work from others that females living with HIV are at a 33% increased risk of death from non-communicable disease. So this is telling us that women are not dying of AIDS and are not dying of infections. This means they're dying, this is cardiovascular disease, liver disease, kidney disease, cancer, which are all considered aging diseases. And so Tanya, when she approached this, she did study many of these aspects in her thesis, but I'm only going to talk about the cardiovascular disease risk. And when we approach that in women living with HIV, we're having to consider various factors. First, all the traditional risk factors that are there for anyone are also present. So we're talking, high blood pressure, diabetes, dyslipidemia, smoking, obesity. Then we have the HIV-specific factors. So the fact that they will often have more latent and chronic infections than people without HIV. They have chronic inflammation, immune activation, and then there's the potential effect of medication, right? If the medication mutate mitochondrial DNA, that could be a factor that's at play. Then you have the factors that would be female-specific. So pregnancies, hormonal fluctuation, and disproportionate stress that are specific to women. And then finally, you have the women living with HIV that have specific factors, such as the fact that they are, the traditional risk factor are typically increased in women living with HIV. So more dyslipidemia, more obesity, more smoking, more everything, more hypertension. And they also have more social structural adversity, meaning they're more likely to have experienced trauma, have experienced violence, have experienced housing insecurity, food insecurity, etc. So in this context, she decided to look at their estimated cardiovascular risk. And to do so, she used a novel estimator or calculator that was developed by the American Heart Association in 2023, and it's called PREVENT. It's a little bit similar to things like the Framingham score that people may have heard about, but it includes more elements and more parameters, and believed to be superior, naturally. And it will give you both a 10-year and 30-year risk estimate, but I'll only talk about 10 years today. And it can estimate your risk of a total cardiovascular events, atherosclerotic cardiovascular events, and heart failure. And this was validated in adults aged 30 to 79. So that was an advantage because most of the other calculators, such as Framingham, only apply to people who are older, 40, some of them 50. This one goes all the way down to 30, allowing us to put more people in our study. And then the last thing that's advantageous with this calculator is that it gives you something called Prevent Risk Age, which is basically a patient-friendly measure that you can use, that clinicians can use in their clinic, which gives you your cardiovascular risk age and whether you can right away tell, is it older or younger than your real age or chronological age. So they have this, so on the right here, I show you the elements that are part of this calculator. Most of them are quite traditional. What's new here is a consideration of eGFR for kidney function, diabetes, and body mass index. So it incorporates more of the kidney cardiometabolic aspects of risk. They have this website. I just thought I would show it, partly because, it's easy to use and it's kind of fun if you know your numbers. But in each of these, you see in pale, there are numbers that are indicated. So these are the range within which you have to be able to use this calculator. So if, for example, you're above 39.9 BMI, you cannot use this calculator. It's not validated for people outside that range, and that because of that, we had to exclude some of our participants. I did mine because I had all my values, and I came up 3 years older than my real age, which was, disappointing, but a bit of a kick in the butt to take care of myself. The study sample selection for this, she started from, as I mentioned, the BCC3 cohort and the CLSA. Within the BCC3, the first part at the top, it shows you everybody that got excluded because they were out of range of the age or BMI, or they were missing data, etc. So she was left with group 2 and group 1. Group 1 being women in BCC3 living with HIV, group 2 being women in BCC3 not living with HIV. And one thing I want to mention is, unlike most cohort studies looking at people living with HIV, we have controls, and we purposely enrolled controls that shared social demographic characteristics with the people living with HIV in our cohort. So we have very similar rates of smoking, very similar income, so that we're really comparing people who have similar lived experience apart from their HIV. And the group 3 is the CLSA, meant to be women, more representative of everybody in the population. And we selected only the ones that were enrolled in BC, and then we matched the CLSA women with the BCC3 women with HIV on a 3-to-1 ratio based on their age and the first 3 digits of their postal code. This is a big table, I apologize, but I wanted to show you primarily all the variables that are included in the PREVENT calculation. So on the first two column, group 2 and group 3, show you how the 2 BCC3 groups, their characteristics and p-values for whether or not they differ. And I hope you can see that they are very similar, except maybe for taking lipid-lowering medication and kidney function. And then you have group 3, the women from the CLSA group, that are then compared to group 1 and group 2. And there you can see a lot more differences. And it's fairly striking when it comes to things like the cholesterol, the blood pressure, and the EGFR and all that are quite different between, as well as tobacco smoking. I want you to note that 30+% of our participants in BCC3 are current smokers, while it's only 3% in the CLSA. Then looking at the social determinants of health. So these are all not in PREVENT, but things that we know may definitely affect aging. So their ethnicity, whether they're living in a rural or urban area, as that may affect how they access healthcare, their level of education, their income, whether they've ever experienced homelessness, whether they have current food insecurity and current substance use. And again, you will see that the 2 groups from BCC3 are fairly similar apart from ethnicity, because indigenous women are overrepresented in the group with HIV, and definitely the Black women are not represented at all in the CLSA study. Again, we see that there are big differences between the CLSA and the BCC3 women with respect to these factors. And unfortunately, the bottom 3 are not data collected in the CLSA. But I'm showing you here Statistics Canada values that I could gather for Canadian women over the age of 40. And one thing I think you could appreciate is that, for example, having an individual annual income less than $20,000 a year, it's over 45% in our BCC3 cohort and only 2% in the CLSA and 10% on average in the Canadian population. So big discrepancies there. Similarly for homelessness, food insecurity, current substance use, the BCC3 cohort is much higher than the Canadian population in general. So this is what she calculated with respect to the 10-year risk of an event. So the first set is for cardiovascular event, total cardiovascular event. The second graph is for atherosclerotic CVD, and a third is for heart failure. And the pattern that emerges here is that the 2 BCC3 groups are very similar with and without HIV, and the CLSA women without HIV fare a lot better. In fact, there's no difference between the 2 BCC3 group and a lot of difference with the CLSA women for each of the 3 measures. And in fact, 25% of the women in the BCC3 cohort fall into the intermediate to high risk group for a cardiovascular event. Then when we went to PREVENT risk age, this is calculating their CVD risk age, I guess. On the left, you have their chronological age, or their real age. And unfortunately, once we had the missing data exclusion and all that, we found that the 3 groups were not perfectly matched for age, but reasonably well. So 54, 56, 54, it was the median age of each of the groups. And on the graph in panel B, we are showing you their calculated prevent risk age. And I think what you can easily see is that both BCC3 group are elevated when the CLSA group is essentially the same. And on the third graph shows you in black the proportion of the women who are more than 10 years older than their chronological age, in red 5 to 10 years older, and in gray who actually are younger than their chronological age, prevent risk age, younger than chronological age, and that is only detected in the CLSA women. So we're seeing a significant age advancement compared to the chronological age in BCC3, but not in the CLSA. So it was an unexpected observation number 3 for us, is that we really did not see HIV having much of an effect. It really suggests that the differences that we see may be associated with non-HIV factors, such as potentially adverse social determinants of health that we know disproportionately affect women in BCC3, and that could potentially explain these differences in cardiovascular risk. The limitations is that BCC3 may not be representative of all women living with HIV. CLSA may under or overrepresent some characteristics, and data on many relevant and important determinants of health were not available in CLSA. But let's say, and this, I'm not presenting it here, but our data suggests that, for example, having ever experienced homelessness is very strongly associated with adverse health outcome. Then maybe this is a question that clinicians can easily incorporate into their clinic with their patients. Not only take a blood pressure, but also ask the question, because in our hands, having experienced homelessness may be a greater predictor of adverse health outcome, including cardiovascular disease, than your blood pressure. So on this note, I want to end by acknowledging this was work that involved a lot of people. Vignette number 1, we want to acknowledge all the COMAPREG and EPIC participants, the study team, investigators, and staff. A lot of the initial work on this was done by Rachel Dunn, who did a master's in my lab. And Haley and Loic helped with the Vignette 1 data. Edward helped with the Vignette 2. Dr. Andre Matman was the clinician who helped us figure out if we should follow up on this participant. And Dr. Laura Sauvé was the pediatrician who connected him for care. And finally, for Vignette 3, this was work mostly by Tetiana Popshedna, but I want to acknowledge all the other people and the funders who supported this work. Happy to take any questions.
Since Ed has to leave, I will moderate the question and answer session, and I welcome any questions for Helen.
Yeah, Ed had to leave.
Yeah. Yeah, please. You can, yeah.
Thank you for the great talk. I have kind of curious about so the HIV, the women might have more higher mutation of the mitochondria. Does it also inherited to the baby as well? And are they also affecting their age and how long they can live?
Did you?
Thank you.
I don't think I heard your question well. Can you speak a little louder?
Oh, okay. So the women with HIV, they have a higher mutation of mitochondria, will affecting their age. Just wondering, does it also affect their baby as well? So they will have higher mutation of...
we hypothesize that. Basically, what we found is not really. We didn't see a difference by HIV. We see differences by other factors, but I'm not present. We didn't present these data here, so yeah.
Helen, is there any known case where the HIV DNA after reverse transcription integrates into mitochondrial DNA?
That's an interesting one. I followed that for a while, and actually a poor student spent an entire summer trying to prove or disprove this. We were told by somebody on campus here that they had detected HIV in mitochondrial DNA, an insertion of HIV in mitochondrial DNA. And then we thought, oh my God, Jack, this could be something, especially that there's mitochondrial DNA in platelets, and then maybe, platelets would transmit the virus. We chased that for quite a while, quite hard. And I think if there is, we could not reproducibly detect it. So we would detect it once, but let's say you extract DNA and you have 100 microliter of DNA, we would do a PCR and then we would see something, but then the next time we tested the same tube, it was not there because it was so rare and so dilute that, and eventually abandon the quest. But it's possible. That would be my answer.
And on the related theme, so how many of the HIV drugs, I guess in the normal cocktails, do affect mitochondria as off targets?
Well,
clinically
the NRTI are what we call the backbone, and usually there'd be 2 out of 3 of the drugs would be NRTI. But more recently, what we're finding is that the new class called integrase inhibitors, they are meant to prevent integration of the HIV into the genome. They are doing something to the mitochondria. Women gain weight unexpectedly on this drug, and in vitro, we see that it affects mitochondrial membrane potential, it affects mitochondrial reactive oxygen species, etc. But the exact mechanism has not been figured out exactly. It could be also that it affects. Mitochondria are involved in hormone production, so it could be through that route as well. I don't know. I don't know if any of you are using human specimen, but this idea of, may I find and may I observe something that could warrant follow-up with the participant who provided the specimen is not something we thought very hard when we said, yeah, we'll put this protocol to ethics. And then when you're faced with having to implement it, it was quite an ordeal. And then for Zesho, this realization that the DNA was not the same based on which lab was providing it caused quite a delay in his PhD, having to follow up what was happening there. Yeah. All right. If there are no more questions, thank you.
Thank you.